Welcome to the Explainer. Today, we're unpacking the ultimate biological assembly manual, human desire. We like to think of love and attraction as pure poetry, but beneath all those feelings is an incredibly sophisticated and honestly sometimes surprising biological machine. Over the next few minutes, we're going to structurally break down this architecture, moving all the way from microscopic hormones acting as chemical messengers, right up to the complex macro-level behaviors that shape exactly who we love and how we identify. Okay, let's dive right into our syllabus. We're going to start at the micro level with the chemical messengers, wire up the autonomic nervous system, look at structural brain differences, explore the neurochemistry of mating, and finally tackle the biology of identity. Section 1. The chemical messengers, building the hormonal foundation. Because, well, before we can even experience a sexual response, our bodies need the raw chemical materials to build and drive the entire system. Our sexual development and our adult behaviors are completely governed by steroid hormones, which, amazingly enough, are actually synthesized from cholesterol. Since they're steroids, they can pass right through cell membranes and directly influence our gene expression. But how does your body know when to release them? It all comes down to the hypothalamic pituitary gonadal axis. It's basically a massive biological chain reaction. It starts in the brain, where the hypothalamus acts as the master gland, releasing a messenger called GnRH. Like the very first domino falling, that triggers the pituitary gland right below it to secrete LH and FSH. Finally, those messengers travel all the way down to the gonads. In males, LH stimulates the production of testosterone, our primary androgen, while FSH aids in sperm maturation. In females, this exact same cascade regulates the menstrual cycle and the secretion of estrogens, like estradiol. It's just a perfectly orchestrated sequential command chain. And this brilliantly illustrates a core concept in biology. Hormones do two entirely different jobs depending on when they show up. First, we have organizational effects. These happen very early in development, primarily before birth, and they permanently wire the circuitry of the brain and the physical genitals in a masculine or feminine direction. Think of this as, well, building the hardware. Then we have activational effects. These happen much later in life, and they're usually temporary. This is when testosterone levels spike in anticipation of mating, or when estrogen levels peak during a female's window of highest fertility. This is essentially the software temporarily running on that hardware we built before birth. Moving on to Section 2, the autonomic nervous system, wiring the sexual response. Let's transition from chemical hormones and see how this builds into the actual electrical wiring of our nervous system. So once that hormonal foundation is set, actual physical arousal requires this absolutely fascinating physiological handoff. To become aroused, resulting in the erection of the penis or clitoris, the body relies entirely on the parasympathetic division. This is your body's rest and digest system. It releases a chemical called nitric oxide, which relaxes the smooth muscles in the arteries so they can engorge with blood. But here is the biological catch, and it's a big one. To complete the sexual response and actually achieve orgasm, your body has to execute a sudden, intense shift over to the sympathetic division, which is your fight or flight system. The sympathetic outflow is what actually triggers emission and ejaculation in men and strong vaginal contractions in women. It is a highly coordinated, incredibly high-stakes relay race between two totally opposing sides of your nervous system. But the physical response isn't just about spinal reflexes or lower body organs, right? Experiencing an orgasm requires massive cerebral integration. In fact, clinical reports, quite surprisingly drawn from epilepsy patients, have shown us that the temporal lobe is a crucial player here. Electrical stimulation of the medial temporal lobe or the basal forebrain can actually evoke profound sexual arousal or even feelings of orgasm entirely from within the brain itself. No physical touch required. Section 3. Structural brain differences, dimorphisms in the anatomy. Now we're zooming out from the nervous system's electrical wiring to look at the macrostructural differences in the male and female body. When biologists look at these structures, they're searching for sexual dimorphisms. These are distinct, measurable structural differences in the brain and body between males and females that correspond directly to different biological needs. It really just comes down to the ultimate rule of biology: form follows function. Let's consider the hypothalamus. Deep inside it, we have a tiny cluster of cells called INH3. In humans, this specific structure is reported to be about twice as large in men as it is in women. In rats, a similar structure can be up to 8 times larger in males, which is wild. Next, moving down to the sacral region of the spinal cord, there's a cluster called Onuf's nucleus. This contains significantly more motor neurons in men. Why? Because it specifically controls the larger bulbocavernosus muscles used to maintain a penile erection. The biology is quite literally physically customized for the necessary mechanical output. Now, what's really interesting here is the profound biological paradox behind how the male brain gets masculinized in the first place. You'd probably assume it's just testosterone flooding the brain, right? Well, actually, in many mammals, for the male brain to be masculinized prenatally, testosterone must enter the brain and physically be converted into an estrogen, specifically estradiol, by an enzyme called aromatase. It's actually this estrogen that triggers the specific patterns of gene expression to masculinize the male nervous system. It's a brilliant biological workaround. Section 4. Neurochemistry of mating, the molecules of love. We've built the hardware, and we've wired the physical response, but what about complex social behaviors? How does the brain regulate things like monogamy and parenting? To understand mating strategies, biologists often turn to a very specific animal model, the vole. We have prairie voles, which are highly monogamous, they mate for life, and they co-parent. Why? Well, because they have an incredibly high density of receptors for two specific neuropeptides, oxytocin and vasopressin, which are located right in the reward centers of their brains. And then we have montane voles. They're closely related, but highly promiscuous, and they don't co-parent at all. The difference? They completely lack these specific receptor patterns. For the prairie vole, bonding with a mate literally triggers a chemical reward in the brain. And it turns out humans really aren't that different. Just look at the data from functional MRI scans of human brains. When people are shown pictures of their romantic partners or their children, it's not just some vague feeling of warmth. Specific reward circuitry literally lights up. And it's driven by those exact same neuropeptides, oxytocin and vasopressin. Specifically, we see dense activity in brain areas like the striatum and the caudate nucleus. So our profound feelings of maternal and romantic love are, chemically speaking, deeply rooted in the exact same ancient reward systems that keep a prairie vole returning to its mate. Which brings us to Section 5, Biology of Identity, Nature, Nurture, and Orientation. How exactly do genetics, hormones, and society mix together to determine our gender identity and sexual orientation? A massive question in biology is whether identity is learned or innate. So the crucial point is understanding the complex interplay of nature and nurture here. A tragic but medically significant case study is David Reimer. Following a severe surgical accident in infancy, David was reassigned and raised entirely as a girl, complete with female socialization and hormone treatments. However, he never identified as female and eventually transitioned back to living as a man. This heavily suggested to the medical community that gender identity isn't just a product of how you're socialized, it's likely deeply built into the brain by those prenatal hormones we talked about earlier. We see similar ongoing research regarding sexual orientation. Neurobiologist Simon LeVay famously studied the INH3 region, that same sexually dimorphic nucleus in the hypothalamus we discussed a few minutes ago. LeVay found that in homosexual men, the INH3 was significantly smaller than in heterosexual men, making its size much closer to that typically found in women. It's a fascinating structural observation, suggesting a potential biological basis for sexual orientation. Impartially speaking, though, it's important to note that these findings remain a subject of active, ongoing scientific debate and haven't always been consistently confirmed across all subsequent studies. Which leaves us staring at a profound frontier in human biology. We've traced the path from cholesterol synthesizing into hormones, through the autonomic handoff of arousal, past the dimorphic structures of the hypothalamus, and right into the oxytocin receptors of our reward system. But it begs an incredible question. If our deepest desires and our identities are such a heavily engineered mix of prenatal hormones and neural wiring, just how much of who we love is dictated by our nature versus nurtured by our experiences? Thank you so much for joining me for this explainer today.